So, our speaker for today is Dr. Anshu Bhardwaj. Dr. Bhardwaj is biologist and data scientist. She obtained her Ph.D. in 2008 in understanding genotype-phenotype correlations in human mitochondrial diseases from CSIR, CCMB Hyderabad. She has over 50 publications to her credit and has trained over 500 young students. She served as an associate scientific advisor to Science Translation Medicine and is on the editorial board of PLOS Global Public Health, Journal of Genetics, review editor for Frontiers in Systems Biology, Protein Bioinformatics and Computational Genomics, and advisor to F1000 Research Galaxy Gateway. Dr. Bhardwaj has received several honors and awards. She was one of the 4 young innovators in India. United National Development Program, selected for the prestigious International Visitor Leadership Program by US State Department, awarded Newton Bhabha Fund from the British Council and the Royal Society of Chemistry UK, and long-term group leader fellowship by CRI Paris. More recently, her lab has developed a unique scientific video game for awareness on AMR, which is endorsed by WHO India. Her passion is towards biomedical big data analytics platform with focus on understanding infection and rare disease biology for better therapeutics and diagnostics. Thank you, Dr. Bhardwaj, for joining us, and the stage is all yours.
Thanks, ACS, for inviting me to this ACS Science Talks, and I'm really looking forward to interacting with all of you and take this as a discussion, because we are dealing with applications of data science in genomics and drug discovery, which I think is not only the current conventional approach to doing science, but it's also futuristic in what it can and how it can impact human lives. So what I have done for today's discussion is I have selected antimicrobial resistance as a topic of today's discussion. The reason of selecting AMR is because we all know it's a silent pandemic. More importantly, AMR is a sustainable development issue. There have been several reports where AMR has been linked to pretty much all SDGs, saying that if AMR is not addressed, a lot of other SDGs will also not be able to deliver, making it a very, very important problem to address. And that is why WHO has declared AMR as one of the top 10 global public health threats. And as well as in India's G20 presidency, both One Health and AMR were the central points of discussion, clearly indicating the fact that this is a global public health emergency, and we all have to collectively work to address it. Now, when do we start? When you go back in history, when was the first time we observed that this kind of phenomenon is happening? Is when there was this correlation between commercial deployment of antibiotics and the emergence of resistance to these antibiotics was observed. We all know AMR is an evolutionary phenomenon, so obviously this was bound to happen, and this is what continues to happen now, making it very, very hard to discover new antibiotics because there are new mechanisms that you see. For some of you who don't understand what AMR is, because it might be a mixed group, I have kept a very simple slide simplifying the concept. So imagine there is an infection and there is a population of bacteria surviving there. So when we take antibiotics and these bacteria are exposed to antibiotics, some of them actually die, and those are the susceptible strains. So those bacteria are killed by the antibiotic. But the ones, as you can see here, the golden color antibiotic has a resistance mechanism. So they actually, in absence of the susceptible strains, these are the ones which proliferate. And now you need antibiotics to kill them. There are many mechanisms of resistance that has been reported in literature, including the fact that bacteria will not allow antibiotics to enter by modulating with their cell membrane. Once the antibiotics have entered, there are mechanisms to throw the antibiotics out or degrade them or modify the target or do a target bypass. Despite the fact that the mechanisms are pretty much same across a large number of bacterial species, the players and the regulation of this is very, very complex. And that is what makes the problem very hard to understand and to address. Apart from the bacterial evolution to antibiotics, there are other reasons of treatment failure that one has to understand. So one of the first is misdiagnosis. We are not treating the right infection with the right antibiotic. There is non-compliance due to several issues in terms of long-term regimens. There are a wide variety of strains that exist in the population. There are wide variety of genetic variability in the host, the human host, and the pharmacokinetics of the drug itself may render it ineffective. Then there are different states of a bacteria, like planktonic versus biofilms. Most of the drug discovery project focus on the planktonic version, whereas 99% of microbial life is biofilm in nature. Then there is drug resistance versus persistence and many more. And a lot of groups in the world, including ours, are trying to see how many of these problems can be tackled and addressed. What all of this AMR has, there has been recent reports in terms of what is the impact of AMR in terms of mortality. And the most recent data indicates that the approximately 5 million people have died from illnesses associated with AMR, and they are killing more people than, the DR infections killed more people than HIV, AIDS, or malaria put together. So the numbers really don't look good, and that is why we need to address this as a collective effort. So the World Health Assembly in 2015 came up with a global action plan to address AMR. And our lab focuses on the first two verticals, which is how do we, through knowledge generation, through research, can contribute, as well as contribute towards awareness of this issue. Over past several years, we have contributed and worked on several projects addressing AMR. But for today's discussion, I have selected 4 stories, which I'll try to explain to you in the most simplistic manner. They are all published, so you can actually go and refer to a published data for more information. And they range from identification of targets to identification and prioritization of potential inhibitors. Let's start with the first one. So for our first study, we selected tuberculosis as the first disease indication for obvious reasons. India is the hardest hit country by TB. It's an infection caused by Mycobacterium tuberculosis, and the multiple drug-resistant TB is a great public health crisis today. And these are reports coming from very latest NTB goal. So our first objective, and the study that I'm going to describe to you is not something very recent, but we started working on TB because even then it was a global public health threat. So we wanted to take a data-driven, unbiased approach to understand the resistance mechanisms of MTB. And we didn't want to go with a very biased, knowledge-based approach. We wanted to take a data-driven knowledge approach so that we are encompassing all possible routes of information flow. And the reason why we did that is holistic understanding is important. There is a very famous story that I'm pretty sure most of you are aware of. So these are 6 blind men trying to understand this elephant, and they don't know what this is. But you can imagine none of them is saying it's an elephant. Somebody is saying it's a wall, somebody is saying it's a rope or a tree or a snake, but they are not able to comprehend that they're looking at an elephant. Now, when I translate the same scenario to how we are looking at data today, it's the same problem. We are saying protein X is important, it's a drug target. We are saying compound Y is a drug. But are we doing our best to basically make that assessment? Are we addressing that, are we able to comprehend the elephant? And that is what we started doing with MTB. And in order to do a data-driven project, first what you need is data. And the data is a lot of data is still sitting in literature. So we started mining this data manually from over 45,000 papers. And this was a huge crowdsourcing activity that we did, which I'm not describing today. But after mining all this data from literature, we were able to generate protein-protein interaction and metabolic map for MTB. These both networks were evaluated in a very graph theoretical approach to identify potential drug targets. What I'm going to do is I'm going to very briefly tell you how did we look at this data, right? Since I'm assuming most of you are chemists, you know how chemical reactions are, visualized. So you have reactants and then products, right? And we wanted a more simpler version of this data so that we can understand what's happening. So instead of looking at data in that linear fashion, we just organized that data where all the metabolites in mycobacteria were listed from top to bottom in one row, all the genes in the second one, and all the reactions in the third one. And what we did is we actually draw a line whenever a gene was responsible for metabolism of a specific reaction, and all the reactants and the products were then linked to that particular enzyme. Then this enzyme and every other enzyme then was linked to the reaction ID, and they were clubbed in different pathways. So this we called as a systems biology spindle map. And all the data that we now curated from literature was represented in this fashion. So when we actually draw this data, this is how much we could curate at that point. And this actually tells us that this is probably the complete metabolic information in mycobacteria. Now, this is what you all have. But is mycobacteria using all this information to grow? And that is when we applied the method of flux balance analysis. And when we actually apply FBA on this data, what we noticed is that only half and sometimes 1/3 of the information is required for mycobacteria to survive in a wild type environment. So it is not even using all the reactions. What are then why it is actually having all those reactions in the genome? So what we did, we now, this is a wild type mycobacteria growing in culture. Now what we did, RB1484 is a target for isoniazid, which is a very potent TB drug. So what we did, we actually knocked out this protein from the network, and what we saw is there was a tremendous information loss, and that is probably why isoniazid works so well for mycobacteria. But what we did in this knockout condition was to look at what are the kind of reactions that came up with a positive flux that had no flux in wild type scenario. And that is when we got these 60 genes which were not really participating, or these 57 reactions which were not showing any flux in the wild type media started showing flux now. And this actually tells us that these are the kind of mechanisms that bacteria is employing towards emergence of mechanisms to deal with drug impact. So what we in principle did, by generating a knockout of a particular route of information flow, we identified other hidden information flow routes in mycobacteria to see what kind of metabolic rerouting is going on. And when we compared this metabolic rerouting data to expression data sets where you actually have exposed mycobacteria to isoniazid, we could identify that 42 of the 60 candidates that we have identified were also shown in the expression data sets, lending credence to our observations and what happens to mycobacteria when metabolic rerouting happens. Now with this and with the other approach that we have not discussed put together, this allowed us to identify several new drug targets and validate a large number of existing drug targets in mycobacteria. Now the problem is you do have targets identified even in literature before. So the next challenge we had is, even when you have targets identified and you do design inhibitors against these targets, target inhibition does not translate into bacterial killing in MTB because it has a very, very impervious cell wall. Nothing really gets inside. So the challenge we have was, even if you have a target, even if you can design inhibitors, it probably never will kill MTB. So the approach we adopted in this scenario is, again, developed a new method where we identified new lead generation strategies for these targets. And I'll give you an example of one target today. And what we did is, once we identified and we designed these new molecules, we then tried to identify which of these new molecules have structural similarity to the ones that are showing whole cell kill in MIC, or which are actually similar compounds. So the approach is that if you have, like our case study starts with a target which has two binding pockets, substrate binding pocket and cofactor binding pocket. And in both the binding pockets, we identified certain interaction features which are unique to these binding pockets and combined those features. So now any new design should have both the features in order to fit in both the binding pockets. Now, in order to test this hypothesis, which we call as this method, hybrid pharmacophore approach, we selected MTB-DAB-B as a target for several reasons, because MTB-DAB-B is involved in several metabolic pathways in mycobacteria. It came up in our metabolic model. It is already validated. There are approximately 9 crystal structures available. So there is a lot of information on the structure of this protein and what kind of monomeric form it has. A lot of information is also available on the binding cavity, where where the PDC or the substrate and the cofactor, which is nicotinamide part of the cofactor, binds. So having this information in hand, what we started doing is we designed a hypothetical molecule. We have no clue whether such molecule will ever exist, but we designed a hypothetical molecule that has binding interactions with at least 2 binding pockets. And then we did our hybrid pharmacophore modeling and searched approximately 15 million compounds from literature, as well as 3,000 compounds that actually have hit in mycobacteria, like we have MIC values. And then we looked at what kind of data this approach is generating. So this is just to tell you how a dynamic hybrid pharmacophore model looks like. So what we did, if you look at this is a nicotinamide part of the NADH. And this is the, the PDC, NADH PDC hybrid. We did the same thing with NADPH also, because this particular enzyme has 2 cofactors and both of them work equally well. So what we did is we generated several such models, where we actually generated the hybrid pharmacophore. We also generated only the cofactor pharmacophores to see how differential these molecules are. I'm not describing the details here, but in simple scenario, we selected like 6 different models, did empty simulations, and then identified certain pharmacophore models for screening against arsenic, zinc libraries, which are mostly drug-like. And then when we identified these NSAID compounds, which are coming from the cofactor interactions versus the HSAID compound, which are coming from the hybrid interaction features, we made 3 observations. First is that the HSAID molecules are structurally very different. So that's a good news, because this is something very promising in a drug discovery project. They are more drug-like than the NSAID. And the third thing is their binding affinities to MPV-DAPV were way better than only the cofactor binding sites. This is some of the data that you might like to review, but just to say that, this is a completely new method. And of 3,000 curated MTB active compounds with MICs reported, for MTB-DAPB, we could have identified 8 compounds for which the mechanism of action is not known, and we believe that they are acting through DAPB. So this is something which we require to, now need to experimentally validate. But just to confirm that as opposed to a lot of conventional methods, our method bypasses a lot of limitations in terms of MTB drug discovery. Though we have tried this only on MTB-DAPB, the method is pretty much applicable to any drug target space. So I know I'm moving fast, but I just wanted to give you ideas in terms of what kind of methods can be developed and what we have done so far. So with that, we have practically finished the first 2 verticals, which is how to identify targets and how do you then identify inhibitors to those targets. In the next 2 stories, I'll be taking a very different approach, which is mostly drug repurposing. And we did that both on small molecules as well as peptides. Why we did drug repurposing is because I have been telling you AMR is a very critical issue. So we need quick solutions. And we all know drug discovery is a very long, risky process with high attrition. And that is where drug repurposing can really help, in the manner that if we in some way identify whether 2 different drugs have similarities with each other, or the targets they act on have similarities, then drug 1 may be repurposed for disease 2 and drug 2 may be repurposed for disease 1. And this is in principle the fact that you are repurposing a drug for an indication other than for which it was originally designed. Now, we have done a book chapter on this a few years ago, because there are several methods with which you can do this. The method that I'm going to focus on today is the one where we have applied a network pharmacology approach. And why we did that, I can tell you the reason. So this is the data coming from Drug Bank. So if you look at the Drug Bank data, and I'm talking of Drug Bank version 4, what we observed is that only 7% interactions in Drug Bank are one target, one drug. Approximately all 93 remaining interactions were either one drug, multiple targets, or one target, multiple drugs, which means there is inherent promiscuity in the way drug targets interact. And this gave us an idea that why not we actually harness this promiscuity and then try and predict what can be repurposed. And the focus here again was TB. And we worked on 49 TB targets which were available in Drug Bank. So very quickly, again, methodological details we can discuss in the Q&A section. But what we did, the first job we did was to enrich the network. So we did a gene ontology based network enrichment. And from 15,000 DTIs available in drug bank, we could enrich the network to approximately 26,000 edges. Then we applied a network-based inference strategy where from known interactions, by applying a very simple two-pass method, we can predict new drug-target interactions. With these new drug-target interactions, we identified which targets this method prioritized, and then we looked at the ones where target has not been shown to have any role in drug resistance, because that was our priority. And then we looked at the drugs that we may shortlist, and some of the predicted drugs were antiviral, antifungal, they were used for bipolar disorder, nutraceuticals, and we are proposing them that they may be repurposed as TB targets. Now, if you notice here in the entire method, we never cared about, we never included the structure of the protein or the small molecule. But when we compared the predicted drug versus the known drug for our candidate proteins, so for some of them, we could find that they were structurally similar. So this means that the method really has identified something very, very interesting. Now, this brings us to the second drug repurposing strategy where we have applied machine learning, but we applied that not on the planktonic forms, but on the biofilm form. And the reason is, for so many years, we worked on planktonic cultures, and then we realized that if 99% of microbial life is biofilm in nature, and we keep developing compounds only for the planktonic version, it might not be the right strategy. So single cells are mostly free-floating entities and responsible for acute infections, and this is a planktonic state, whereas biofilms are responsible for the chronic infections, and they are sessile communities, so they are actually not mobile. And the fact is that they are very, very old, so they are more than 3 billion years old, and residents of the biofilm are up to 1,000 times more resistant to antibiotics than the planktonic cultures. The other thing is, where do you find them? You practically find them in all types of hospital infections, whether they are urinary tract infections, or nosocomial infections, or blood infections, or pneumonia. So the thing is, it's everywhere, and they're very, very difficult to get rid of. How does a biofilm look like? So if you look at this microcolony structure, I have shown one here. So in principle, the colonies that exist in nature are polymicrobial. So there are more than one type of organism staying there. And if you see, there is a gradient of yellow that I'm showing here, which tells you that there is a difference in the distribution of species. Ones that are on the outer layer of biofilms are metabolically more active and hence can be targeted by fluoroquinolones, whereas the ones in the central part of the biofilm are very, very slow growing, and that is where you need to have lipopeptides. So they are very difficult to harness because they're very heterogeneous in nature. And the catch is that the strategies that biofilms adopt for AMR are different from the planktonic versions, because here the issue is that you actually have a physical barrier, first of all. Second, there are more than one microbes present, so they can actually share a lot of genetic material among each other, making treatment of biofilms extremely hard. And a lot of enzymes that can degrade antibiotics synthesized by all of them are floating around in the EPS. So the matrix is very, very inhabitable for the antibiotic to survive. So how do you find solutions? So we identified what nature might have done to do this. So we noticed that in our system, there are a set of peptides which are called as catalycitins, which actually can disrupt biofilm structures. And that gave us an idea that why not, instead of looking at small molecules, look at peptides that can disrupt the biofilm structure. And we came across this database of BAMs, and they actually had a list of antibiofilm peptides killing all the stages of a biofilm life cycle. Now, what we did here was a little different what we normally do in a machine learning project. The positive data set in this project were the anti-biofilm peptides. So they were disrupting the biofilm. The negative data set in this scenario were the the peptides that were maintaining the biofilm. So this is something we took from QSP-PRED. So now this actually made the entire scenario very contrasting, and we wanted to see, just on a sequence basis, are there any differences in them? And we labeled this data, both antibiofilm peptides and not antibiofilm peptides, and generated some machine learning predictive models. and then tested it on independent data sets. And we didn't do one model, we actually developed 6, because we always believe in the ensemble strategy, because one method can be misleading. So we actually did a large number of methods, and I'll not be describing all of them today, but just to tell you that all of these methods are only so far sequence based. There is no structural information on the peptide in these models. If you look at the top table here and accuracies, you can see that the best models are doing a pretty much good prediction on the training data set, but they performed even better on the independent data set, which was a very good news for us. And now we wanted to understand what is that the machine learning model is seeing to make such good classifications. What we noticed is that the antibiofilm peptides were abundant in cationic residues. They were possessing a greater net charge than quorum sensing peptides, and they are hydrophilic in nature. Now, how is this property helping them to function better as antibiofilm peptides? Let's look at the example. Now, the biofilm is mostly water, like it's 85% water, and then you have carbohydrates and extracellular DNA and other things. But if you really see, the EPS matrix has a net negative charge. Now, if you talk of antibiotics like tobramycin, they have a positive charge, and via some unknown ionic interaction, because we don't know what it interacts with, but they get sequestered in the biofilm, so they are not able to function. Likewise, aminoglycosides, the problem with them is they are negatively charged. And since the EPS is also negatively charged, obviously they're not going any deeper in the biofilm. However, antibiotics like ciprofloxacin, which are neutral, they can penetrate deeper. And that is also the case with the peptides that we are seeing. And this is what we think is what the ML models are also predicting, that it's the nature of these peptides which is making them diffuse through the EPS and making them function as they're supposed to, because they're hydrophilic in nature. Now, subsequently, we wanted to see whether these predictions have any value in a real life scenario. So we actually took all these 6 models and made prediction on FDA approved peptide drugs, as well as antimicrobial peptides in clinical trials. And what we noticed is that the original indication of these AMPs, like say, for example, RDS or catheter infections, or if you can talk of immunocompromised patients or oral candidiasis, middle ear infection, they all are primarily sites of biofilm formation, which makes it a tantalizing possibility that needs further testing. We haven't done that, but a lot of groups in the world have used our application and they have synthesized peptides, and they are able to validate a lot of data that the server predicts. So this is a method that is available online for you to use. It's an ensemble machine learning approach to predict antibiofilm peptides. And one other thing you can do is, if your peptide is coming inactive, there is a facility to keep generating mutants of the peptide till the time you can make it active. So yeah, so try and use the server, and you will look forward to receiving your comments on this. Now, apart from all of this work that I described to you today very, very quickly, we also developed a very large number of data analysis platforms for AMR. A most recently published application is ASSIST. So we do a lot of genotype-phenotype correlations in lab, and we realize that there is a lot of challenge in terms of implementation of standards in AST data sets. And that is why ASSIST was developed, and we do machine learning models with this data. So there's a lot of work going on in the lab, and maybe some other time I would be happy to discuss with you what kind of work we are doing in terms of landscaping AMR mechanisms in a very wide range of organisms. Apart from doing all in silico work, we also try and make effort to understand the AMR pathogens in the environment. So this is a very recent report where we have actually isolated an MDR, Acinetobacter baumannii, from air in a market setting in India. So this is something very, very, so we are yet to understand what the source of this MDR, Acinetobacter baumannii is, because it's not clinical. It is coming from air. So is it really the the respiratory thing that is coming, or it's coming from the environment? We are actually now scaling up this study to really map it. But this study told us, or gave us the observations that the AB strains, the Acinetobacter baumannii strains in India, are highly variable. So they are actually, their core and accessory genomes are not what we see in the rest of the world. So this definitely warrants more in-depth studies, and we are at it. Now, with that, we pretty much concluded the science part of the work, in the sense that what we do in terms of developing resources. Going forward, as I told you, we also are interested in generating awareness on AMR, which we think is a very important aspect of science. And how do we make sure that the communities are aligned, because AMR is a very complex problem to solve. So I'm pretty sure all of you have read Sir Alexander Fleming's Nobel laureate speech, and he in that speech very clearly mentioned that if we expose pathogens to antibiotic concentrations which are not enough to kill them, there is a possibility of emergence of resistance. And this is something that he told in 1945, and we, as a the entire world, ignored the point that it's very, very important that judicious use of antibiotics should be followed. Now, subsequent to that, what has happened in past 10-12 years is, I'm showing you only the most recent data, but there are other studies also where people are indicating that till the time the public is aware about the problem of AMR, no amount of new drug discovery or understanding the mechanism is going to address the problem at a pace at which we want it to be addressed, to really stop the impact of AMR becoming even more worse. So what we thought is, why not it's very difficult to influence adults. You all probably agree with that. So we thought, why not influence young children. So we started working with young children to understand what they really like doing. And then we thought, okay, let's do one thing, create something that is more interesting for them to learn from. And that is why we generated a video game, which is called as ARMOR. And it is to raise awareness on AMR. This is currently endorsed by WHO. What I'm going to do is, instead of telling you what the game is about, there is a teaser on the game, and I'm going to play that right now for you. So just 1 second. I hope you can hear the sound. So what we actually did, it's a multi-level game, so that there is interest for the students to come back and replay. Appeal is very important for learning. What we are currently doing is, we are making ARMOR a multiplayer game, so 2 players can really be online, and that is something that is under development, and hopefully in some time you'll be able to play that too. So we entered ARMOR into the International Educational Games competition in ECGBL in 2021, and we were one of the top 5 panelists, like the finalist in this gaming competition. And it's very important why we keep doing this, because at the end of the day, the point is to make the entire society aware of these issues, and that is what we intensely do in the lab, apart from the scientific projects that we do. Since not everybody is a gamer, I'm a hardcore gamer, but not everybody is like that. So we are using other methods, like, say, for example, we are generating now a web series on microbiology, infection, and AMR. The 3 episodes are already done, and this is going to be a web series on AMR that will be available through the Jigyasa platform, free for everybody to watch. And we wanted different ways of engagement. Then there are other things that we do, which is more physical. So we have adopted school in and around Chandigarh, and with our PhD students and with other faculty staff members, we actually go work with them, conduct very simple experiments on microbiology, hand hygiene, and we do play test with the game. And we try and see how much awareness can be generated among the younger generation so that they understand the science behind the game. Now, I have not told you a lot about the game, but the game might look like a gamification, but each element there, the backend, the scientific, the mathematical equation that is built in for the game to respond is all hardcore science. So it is not only game. There's a science behind each level, and that is what we discuss with students when we go for playtest. Apart from this, what we have very recently have done is we have launched the Epic Hackathon. Now, this is a One Health Epic Hackathon, and I would request all of you to take a look at it. This is open for school children to register or send their entries if they have identified solutions to the problems we have focused on in the One Health agenda. So I propose the One Health agenda to be understood by students and to recommend solutions, because once they understand the problem even more clearly, we'll actually have more champions of change in the society. So what I have done today is I have gone through extremely quickly on a lot of resources and research that we do on AMR, but my lab also works on mitochondrial diseases. And that's the host part of the story, the host pathogen interactions and the mitochondrial diseases as such, because mitochondria is one of the primary targets of a lot of pathogens. These are a lot of resources my group has developed in past several years, but I will not have the time to discuss with you. Just what I wanted to briefly convey is that I work with NIH and a lot of clinicians on this point, because that is where I think the impact will really come, and that is what we are currently doing with AMR also. Working with clinicians is important because then you know what the problems that they are facing and how you as a researcher can help them understand it. So this is what I do both for AMR as well as for mitochondrial diseases. The other thing for the audience, I'm pretty sure there must be students in the people who have joined today. This is a paper I would strongly recommend you to read. This is pretty much, past 20 years of human genome to human pangenome. Read this paper. It's going to tell you how we can use genomics as a method for precision medicine going forward. And this is not something which is focused only on infections, but it will cover all aspects of infection, basically communicable and non-communicable diseases. And that's why I wanted to put it here. To close, all this work is not possible without a large community behind it. And I'm very, very happy to tell you that I work with a very large number of undergrads and, postgrads, PhD students, postdocs. But I also work with school children, and these two are like my favorite for the reason they are very, very young when they publish their first papers from the lab. So if I briefly tell you that this DPAP, the algorithm, the machine learning algorithm that I told you, was developed by Pooja Gupta when she was finishing her school. Ashma developed, basically wrote a book chapter with me when she was just finishing her 10th grade. So, just to convey to some of the young students out there, you don't need degrees to do science or age. Passion is what will drive you forward, and these kids have done that. This is just to basically tell you that we are very active on print and online media because we wanted to make sure that science should reach masses, because if you don't do that, people will not trust what you're doing. And COVID taught us one thing, that how do you trust scientists? If you don't know what they're doing, how do you know it to trust or not? So this is very, very important. Towards the end, I would like to say that I'm very, very proud to be part of an organization which has contributed a very large number of resources in terms of databases in biology, and we are among top 4 institutes in the world in line with EBI and NCBI who have contributed these resources, and I'm very, very proud that we do this as an institutional effort, not just my lab specific effort. And then, of course, I thank my lab here in India and in France, and a lot of funding agencies, both national and international, who have supported us all these years. And again, thanking ACS for having me today here. And I'm looking for a lot of people. So if you are interested in working with us, please do approach us. And with that, thank you, and I look forward to taking questions.